Haematopoiesis is the intricate and vital process by which our bodies generate blood cells. This biological marvel occurs within the bone marrow, a spongy tissue residing within the cavities of our bones. Here, amidst a complex microenvironment of stromal cells, cytokines, and growth factors, hematopoietic stem cells, or HSCs, reside, possessing the remarkable ability to self-renew and differentiate into all lineages of blood cells. HSCs are the cornerstone of hematopoiesis, representing a rare population of cells with the potential to give rise to the diverse array of blood cells circulating throughout our bodies. These lineages include erythrocytes, the oxygen-carrying workhorses, leukocytes, the vigilant sentinels of the immune system, and thrombocytes, also known as platelets, crucial for blood clotting and wound healing. The process of hematopoiesis is exquisitely regulated, ensuring a constant supply of mature blood cells to meet the body's demands. Disruptions in this delicate balance, whether due to genetic mutations, nutritional deficiencies, or infections, can lead to a range of hematological disorders, underscoring the critical importance of this process for human health. Understanding hematopoiesis is fundamental for medical students and healthcare professionals alike, providing a framework for comprehending the intricacies of blood cell formation and the pathogenesis of numerous diseases. In the following sections, we will delve deeper into the specific processes of erythropoiesis, leucopoiesis, and thrombopoiesis, exploring the molecular mechanisms, cellular players, and clinical significance of each. Erythropoiesis, the formation of red blood cells, stands as a cornerstone of hematopoiesis, ensuring a constant supply of these oxygen-carrying cells essential for life. This intricate process takes place within the bone marrow, a dynamic microenvironment where hematopoietic stem cells embark on a remarkable journey of differentiation. The journey begins with the commitment of HSCs to the erythroid lineage, a process driven by a complex interplay of transcription factors and signaling pathways. These committed cells, known as erythroblasts, undergo a series of well-defined morphological and functional changes as they mature into erythrocytes. Central to the regulation of erythropoiesis is erythropoietin, or EPO, a hormone primarily produced by the kidneys in response to low oxygen levels in the blood. EPO acts as a potent stimulus, promoting the survival, proliferation, and differentiation of erythroid progenitor cells, thereby increasing the production of red blood cells. As erythroblasts mature, they undergo a series of characteristic changes, including a decrease in cell size, condensation of the nucleus, and the accumulation of hemoglobin, the iron-containing protein responsible for oxygen transport. The final stage of maturation involves the expulsion of the nucleus, giving rise to the distinctive biconcave shape of mature erythrocytes, ideally suited for their role in gas exchange. The journey from a committed erythroblast to a mature erythrocyte is marked by distinct morphological and biochemical changes, reflecting the cell's progressive specialization for oxygen transport. These stages, identifiable under the microscope, provide valuable insights into the dynamics of erythropoiesis. The earliest recognizable erythroid precursor is the proerythroblast, a large cell with a high nucleus-to-cytoplasm ratio, basophilic cytoplasm, and prominent nucleoli, reflecting its active protein synthesis. As the proerythroblast differentiates, it gives rise to the basophilic erythroblast, characterized by intensely basophilic cytoplasm due to abundant ribosomes synthesizing hemoglobin. Further maturation leads to the polychromatic erythroblast, named for its multicolored cytoplasm, reflecting the increasing accumulation of hemoglobin, which stains pink, alongside residual RNA, which stains blue. The nucleus continues to condense and the cell size gradually decreases. The penultimate stage is the orthochromatic erythroblast, characterized by a small, dense nucleus and a predominantly pink cytoplasm due to abundant hemoglobin. Finally, the orthochromatic erythroblast expels its nucleus, resulting in the formation of a reticulocyte, a young red blood cell containing residual RNA. Reticulocytes are released from the bone marrow into the bloodstream, where they mature into erythrocytes within 1 to 2 days. Leucopoiesis, the production of white blood cells, is a critical arm of hematopoiesis, responsible for generating the diverse array of leukocytes that form the cornerstone of our immune system. These cellular guardians patrol the body, defending us against invading pathogens, clearing cellular debris, and maintaining tissue homeostasis. Unlike erythrocytes, which constitute a single cell type, leukocytes encompass a heterogeneous group of cells, each with specialized functions in immune defense. These include neutrophils, eosinophils, basophils, lymphocytes, and monocytes, each originating from a common myeloid progenitor cell within the bone marrow. The differentiation of leukocytes is a tightly regulated process, orchestrated by a complex interplay of cytokines, growth factors, and transcription factors. These molecular signals guide the development of specific leukocyte lineages, ensuring an appropriate balance of immune cells to effectively combat a wide range of threats. Dysregulation of leucopoiesis, whether due to genetic defects, infections, or autoimmune disorders, can have profound consequences for human health, leading to immunodeficiency, autoimmunity, or malignancy. Understanding the intricacies of leucopoiesis is therefore crucial for diagnosing and treating a wide range of hematological and immunological disorders. Section 5. The granulocyte series, neutrophils, eosinophils, and basophils. The granulocytes, comprising neutrophils, eosinophils, and basophils, represent a key component of the innate immune system, providing rapid and nonspecific defense against invading pathogens. These cells are characterized by the presence of granules in their cytoplasm, each containing a unique repertoire of enzymes and antimicrobial proteins. Neutrophils, the most abundant type of white blood cell, are the first responders to infection, phagocytosing and destroying bacteria and fungi. Their granules contain potent enzymes like lysozyme, myeloperoxidase, and proteases, which effectively neutralize pathogens. Eosinophils play a crucial role in parasitic infections and allergic reactions. Their granules contain major basic protein, eosinophil cationic protein, and eosinophil peroxidase, which are toxic to parasites and contribute to the inflammatory response in allergic reactions. Basophils, the least common type of granulocyte, are involved in allergic and inflammatory responses. Their granules contain histamine, heparin, and other mediators that contribute to vasodilation, increased vascular permeability, and bronchoconstriction. The production and function of granulocytes are tightly regulated, ensuring an appropriate immune response to infections and other challenges. Dysregulation of granulocyte production or function can lead to recurrent infections, allergic diseases, or autoimmune disorders. Section 6. The agranulocyte series, lymphocytes and monocytes. The agranulocytes, comprising lymphocytes and monocytes, are essential components of the adaptive immune response, providing long-lasting immunity against specific pathogens. Unlike granulocytes, agranulocytes lack prominent cytoplasmic granules and are characterized by their large, round nuclei. Lymphocytes, the key players in adaptive immunity, are further divided into B cells and T cells. B cells, upon activation, differentiate into plasma cells that produce antibodies, highly specific proteins that bind to and neutralize pathogens. T cells, on the other hand, directly kill infected cells or orchestrate the immune response by releasing cytokines. Monocytes, upon entering tissues, differentiate into macrophages, phagocytic cells that engulf and destroy pathogens, cellular debris, and apoptotic cells. Macrophages also play a crucial role in antigen presentation, a process by which they display fragments of pathogens on their surface to activate T cells, initiating an adaptive immune response. The development and function of lymphocytes and monocytes are tightly regulated to ensure an effective and balanced immune response. Dysregulation of a granulocyte production or function can lead to immunodeficiency, autoimmunity, or malignancy, highlighting their critical role in maintaining human health. Section 7. Thrombopoiesis, Orchestrating Hemostasis. Thrombopoiesis, the production of platelets, is a critical process ensuring the integrity of the vascular system and preventing excessive bleeding. Platelets, also known as thrombocytes, are small enucleated cell fragments derived from megakaryocytes, giant cells residing within the bone marrow. The journey of platelet production begins with the commitment of hematopoietic stem cells to the megakaryocytic lineage, a process driven by thrombopoietin, or TPO, a hormone primarily produced by the liver. TPO stimulates the proliferation and differentiation of megakaryocyte progenitors, ultimately leading to the formation of mature megakaryocytes. Megakaryocytes, as their name suggests, are giant cells with a lobulated nucleus and abundant cytoplasm. They undergo a unique process called endomitosis, wherein they replicate their DNA without dividing, resulting in polyploidy, with some megakaryocytes containing up to 64 copies of the genome. The massive cytoplasm of megakaryocytes undergoes fragmentation, giving rise to thousands of platelets, each enclosed by a membrane and packed with granules containing clotting factors, growth factors, and other mediators essential for hemostasis. Platelets are then released into the bloodstream, where they circulate and play a vital role in preventing and stopping bleeding. Section 8. Megakaryocytes, the giant cells behind platelet production. Megakaryocytes, the colossal residents of the bone marrow, stand as the cellular factories responsible for producing the tiny yet mighty platelets, essential for hemostasis. Their unique biology and intricate differentiation process highlight the remarkable specialization within the Haematopoietic System. The development of megakaryocytes is a tightly regulated process, orchestrated by a complex interplay of transcription factors, cytokines, and growth factors. Thrombopoietin, or TPO, the key regulator of thrombopoiesis, plays a central role in promoting the proliferation, differentiation, and maturation of megakaryocytes. As megakaryocytes mature, they undergo a series of remarkable morphological changes, including endomitosis, a process of DNA replication without cell division. This results in polyploidy, with mature megakaryocytes containing multiple copies of the genome, providing the cellular machinery for massive protein synthesis. The cytoplasm of megakaryocytes undergoes extensive development, forming a complex network of membranes, granules, and organelles. These granules, upon platelet activation, release a plethora of factors, including clotting factors, growth factors, chemokines, and adhesion molecules, orchestrating the hemostatic response at sites of vascular injury. Section 9. The crucial role of haematopoiesis in health and disease. Haematopoiesis, the intricate process of blood cell formation, is essential for life, ensuring a constant supply of erythrocytes to transport oxygen, leukocytes to combat infections, and platelets to maintain hemostasis. Disruptions in this delicate balance can have profound consequences for human health, leading to a wide range of hematological disorders. Anemia, a common consequence of impaired erythropoiesis, arises from a deficiency of red blood cells or hemoglobin, leading to reduced oxygen-carrying capacity of the blood. This can manifest as fatigue, weakness, shortness of breath, and pallor. Leukemias, cancers of the blood and bone marrow, are characterized by the uncontrolled proliferation of immature white blood cells, disrupting normal hematopoiesis and impairing immune function. Lymphomas, on the other hand, are cancers that arise from lymphocytes, affecting the adaptive immune response. Platelet disorders can lead to bleeding diatheses, characterized by excessive bleeding or bruising. Thrombocytopenia, a deficiency of platelets, can be caused by decreased production, increased destruction, or sequestration of platelets. Understanding the intricacies of haematopoiesis, including the processes of erythropoiesis, leucopoiesis, and thrombopoiesis, is crucial for diagnosing and treating a wide range of hematological disorders, highlighting the importance of this fundamental biological process for human health.